Hello. Hi, James.
Hi there. How you doing?
Okay, great.
Good.
Hi, John.
Hi, how are you? Sorry for the big headphones.
Bigger the better. Hi, Anita. Welcome. So this is the first time I'm doing this. So there will be a little bit of a learning. Probably, several others for you as well. But we'll try to see how it goes. Hi, James.
Hi there. How you doing? Good.
Okay, great.
Good.
Hi, John.
Hi. Sorry for the big headphones.
Oh, there's a delay. Someone is playing back.
Huh.
So this is the first time I'm doing this. So there will be a little bit of a learning. It is coming from Leora. Okay, I muted Leora. Oh my God.
So, James, I'm sorry, Emek, what is coming from me? Is there an echo
Hi,
coming
John.
from me?
Hi. Sorry for the big headphones.
Oh, there's a delay. Someone is playing back.
So, Emek, is there a delay coming from
This
me?
is the first time I'm doing this. It is coming from Leora. Okay, I muted Leora. Okay, this was strange. Okay, this is strange. Let's see if it gets any better. Hi, Gully. Okay, I guess anytime someone joins, we are going to hear this sequence. Okay, let's give it a try and see if it works. Okay, I muted actually everybody. So if you want to speak, you need to kind of unmute yourself. There's some echo thing that is going on. I'm not sure why. Hopefully, that won't be a big problem. So I'll share this presentation. I don't know if you can see it. So can you nod your head if you can see it? No? Okay, can you click on the drive link? Or the other thing I can do is I can actually share the desktop. And let me do this. That might be easier. Okay, any better? Okay, so let's go. I'll actually try unmuting everybody back, and then we can actually might try a conversation. Okay, so you already have seen this picture. So this is how the
Any better?
Okay. You have seen this picture, and this is how the BioPAX in SPGN looks like. Okay, for some reason I'm getting this feedback from Leora, I think. So I'll meet her and then we'll see. So these reactions are essentially the center of the BioPAX, and they're extension of the biochemical paradigm. So this is how it looks like. This AKT1 that is blue on the top, it is a protein.
I don't know. Oh, there it is. Okay, finally it updated.
Okay, sorry. This AKT1 is a blue protein that belongs to the AKT1 protein reference RAKT1, and we'll talk about this. I'm just kind of giving you an outline of how things looks like. So here below you have a complex, and it has the members of HSP90 and AKT1. You have different enzyme catalyzing different reactions, and you have different phosphorylation sites. What is not shown here is subcellular location. So this is essentially the type of thing that we're trying to capture. Although BioPAX can capture more, this is going to be the backbone of what we're trying to capture. Okay, so these molecules that you see here, for example, different states of AKT1 in BioPAX, each of these guys will be a different physical entity. Specifically, they will be a subclass of physical entity called protein. Okay, and we have 6 subclasses of the physical entity: protein, DNA, RNA, small molecule, DNA region, or RNA region. There's also a complex, which is essentially for capturing things like this, the molecular complexes. Okay, and then you have for each physical entity, you can describe its subcellular location. You can describe its modifications to the sequence. Also, you can describe molecular complexes and binding. Okay, just a procedural comment. At any point, you can interrupt and then ask questions. So please do. For each physical entity, we have multiple entity references. For multiple physical entities, we have a single entity reference. So AKT, as we know in the AKT1, as we know in the as a uniprot entry, is the entity reference in the BioPAX world. And for different phosphorylated phosphorylated state of AKT1 will be a protein. So you have the protein reference and the protein, and how it looks like is that you have the protein reference, which maps to multiple physical entities, and each physical entity can participate in one or more interactions. Okay? Any questions so far? Okay, so interactions, there are 5 major subclasses of interactions. 2 of them are genetic interactions and molecular interactions, and I won't be talking about those because I don't think that for the purpose of big mechanism they're very important. Molecular interactions and genetic interactions typically in BioPAX come from PSIMI sources, and they are typically captured through high throughput techniques. although there are also interaction databases that are curated, like HPRD. We will be focusing on conversions and controls, and also there's old guy here, which is the template reaction, and I'll briefly talk about this as well. For the conversions, you have each conversion has a set of left participants and right participants. We don't use the word input and output. We avoid that wording particularly because conversions in BioPAX can be reversible. So you also get to define the direction of the conversion. It can be left to right, right to left. It can be reversible, and also we differentiate between the reactions that are physiologically reversible, in the sense that in a physiological context there's always a certain direction, versus the reactions that are truly irreversible. And each conversion has, you can define the stoichiometry of the conversion through a specific utility object called stoichiometry. And also for the controls, you can define the type and direction of the catalytic activity. The conversions also has facilities to represent the entropy, delta G entropy difference, and then the K prime. The BioPAX cannot represent rate constants and rate laws. That was done, intentionally, because we thought that it was not a good idea for integration use cases. It's a long discussion, and maybe we can use some time in this hangout to discuss that. But that is something that we won't be able to represent in BioPAX. So if there's some sort of rate constants and rate laws that can be extracted from the literature in this program, we either need to extend the BioPAX or do something else in parallel to be able to capture those. Any questions? Okay, so there are 4 subclasses of conversion, and they typically have the same behavior. They only matter for validation purposes, but for what it's worth, the 4 types are biochemical reaction, complex assembly, degradation, for example, for representing things like degradation of a protein by proteasome, and then transport for representing events that contain subcellular location changes. Okay. So Anita, for your question, this call is being recorded and it's automatically being streamed in YouTube. So you can swap places with someone if you want to, but they can also kind of watch it from the YouTube. What they won't be able to do is they won't be able to ask questions, I think. Is that fine, or?
Yeah, that's great, thanks. I just wasn't sure. There are a couple of other people who want to join from our team, and I would love them to, because this is incredibly useful. But
Okay.
I think I'll leave and I'll have Gully join.
Okay, sorry about that.
And I'll just...
I didn't realize that
Thanks.
we would max out at 8 people, but I guess this is what it is.
Yeah, I think you probably have quite a few people who would like to join. So anyway.
Okay, I mean, they will still be able to watch it over the YouTube stream, I think.
Thanks.
Okay. So the control subclasses, so each conversion can be controlled by one or more controls, and this is for representing the like events like catalysis, and one interesting thing about BioPAX is that the controls can be chained, so that you can actually have, for example, a small molecule modulating an enzyme that is catalyzing a reaction, and this can be used to represent arbitrarily complex control structures in BioPAX. So now we are getting a little bit into the details. In this case, okay, hi Larissa.
Hi.
So it turns out that there's a maximum number of participants 8 participants. So I guess Anta left, and you'll be able to join in. So anybody, if you feel like watching through the YouTube stream, you can also do that.
Okay, I can do that.
No, you can stay. Oh, okay. Anyway, so as I said, there will be some learning process, and I think this is happening. So as for the entity features, as you can see, there are three major types: the modification feature, binding feature, and the fragment feature. Modification features for capturing things like phosphorylation or acetylation, basically any type of small chemical group addition to the protein or the gene or the DNA, RNA. Binding feature is for capturing like domain binding between proteins, and then the fragment feature is for capturing splicing. For example, when you're trying to represent the creation of insulin from proinsulin, you would like to represent those excised out protein fragments, or if you want to create, the acetylation, basically any type of small chemical. Gali, I need to kind of mute you because there's an echo. So fragment feature allows you to represent things like spliced mRNA or as I said, insulin from proinsulin. And for each entity feature, what you can represent is that you can represent its location. You can represent its location type. So if that feature is happening on a promoter, you can represent that using the controlled vocabularies, which I will describe a little bit later. And then you can define the modification type. For example, if the added group is a phosphorylation, you can represent that again using a controlled vocabulary. One thing that is really important, and especially important for rule-based modeling, is that in BioPAX, if all of the features that are represented are assumed to be present, and all of the non-mentioned features are assumed to be unknown, as opposed to not modified. So in BioPAX, you can actually represent. There are two fields, which is feature and then another field on the physical entity, not feature. So you can actually represent, for example, unphosphorylated a protein that is unphosphorylated at position 21. This is slightly different than not mentioning it at all. In this case, you're saying that you don't know if that protein is phosphorylated at this position or not. So this the problem for some of you, the problem that we are trying to address is obvious, but for the others who are not aware of this subtle but important detail is proteins have typically more than one, sometimes tens of phosphorylation sites, and at a particular biological experiment, we only observe a subset of it. Typically, we only observe a single phosphorylation site. It actually is very difficult to observe double phosphorylation. This goes for other modification features as well. So as you can imagine, if a protein has 10 phosphorylation sites, for example, P53 has 7, 8 functional phosphorylation sites. This leads to combinatorially many states of P53.
Can I ask a question?
Sure.
Can you explicitly mark that a site is unphosphorylated, and how would one do that?
There is a property called not feature in BioPAX. So you basically say P53 protein not feature phosphorylation at 21. Does it make sense?
Yes, thank you.
So that is, yeah. So basically, we have a negative and positive way of saying, of mentioning a feature, and if you don't mention that feature, it basically means an unknown or unspecified. So this way, we actually should be able to capture things in kappa and pathway logic, basically all of the modeling languages that are based on the delta. So in this case, for example, in this first reaction, the delta is P21, the phosphorylation at 21, and for example, kappa and pathway logic uses that delta for representing the event. BioPAX can still represent the delta, but it represents it using a biochemical paradigm, which I think can be converted into the kappa or pathway logic. So another thing that is related is how we represent binding in BioPAX. So basically, we have the binding features they come in pairs. So for example, if you have p53 binding to MDM2, you have a binding feature for p53, and then you have another binding feature for MDM2, and these two guys kind of know each other through a symmetric relationship. And what you can do is you can first create this binding feature, and then you can have the p53 not have that feature, which means that it is an unbound form from MDM2. So this again helps address the combinatorial complexity and allows us to represent things that, for example, you can represent in kappa or pathway logic. One word of caution, though, there's no reason why we won't be able to mine these from the literature. But if you want to use the existing pathway resources for these type of relationships, simply they don't exist. I mean, we put the facility into the BioPAX, but no pathway database was kind of ready to use this feature. So we don't have a lot of prior information that contains not feature.
Question, do you specify the site of the binding between two proteins, let's say between p53? Is there like a named site or anything like that, like there would be in kappa, for example?
Yeah, you can name the site, or you can also specifically define the binding interface through defining the sequence fragments. You can't define a surface or anything like that. You can't do 3D. You can't do that type of thing. But you can definitely, for example, if you have an SH2 domain at a particular site, you can definitely define the end and the beginning points of that. Does that answer your question? Or okay, awesome. So finally, the third thing that is kind of complicated about BioPAX is the so-called generics. And I am sure that those of you who either worked with the biological text before or looked at some of the examples that that came in the starting kit realize that a lot of these proteins or small molecules, for that matter, are not single chemical species, but they are families. So, for example, the Wnt protein, I believe there are more than 10 Wnt's in the human. And they also make a complex with the family of frizzled proteins. So you actually have this combinatorial complexity, and sometimes you just want to talk about Wnt frizzled complex in general terms. So in BioPAX there are two ways of doing this. One is we can define groupings at the entity reference level, and we can define another set of groupings at the physical entity level. Different pathway databases use different methods. Reactome exclusively use the PE-based grouping, while NCI, I believe, used a mixed method. And then all other pathway, or Panther, for example, is exclusively ER-based grouping. This might be a little bit confusing, but I'll try to give an example and kind of expand on that. So in general, we prefer the ER-based method because it is less vulnerable to combinatorial expansion. But there are many cases where the ER-based method might not be possible. And the PE-based method is more flexible, but as I mentioned, it can combinatorially expand. So here's an example of the ER-based method. Here you have on the right side, you have the entity reference for Wnt. And you have Wnt1 and Wnt2 as its family members, and from this generic entity reference rWnt, you create a physical entity. And now through this link through the rWnt and from rWnt to rWnt1 and rWnt2, we know that this Wnt can mean a protein of any of these can be instantiated by Wnt1 and Wnt2. So why is this important? Because you can now talk about generic phosphorylated forms of Wnt, for example. Okay, so if you create a phosphorylated form that belongs to this generic entity reference, then you can actually also show that this maps to certain phosphorylated forms in certain instances of the Wnt family. To be able to do this, you also need to kind of create a nesting or grouping relations for the entity features, and BioPAX allows you to do that as well. So some of the pathway databases like NCI-PID uses that type of information to robustly represent generics. The Reactome way of doing this is you completely remove, you basically kind of don't do anything on the entity reference side, and then for each different state of Wnt, generic Wnt, you map it to a set of specific physical entities rather than specifying it explicitly. Is this clear, or was it too complicated, or no?
So in this example that you're showing here, the generic Wnt has these two sites. Does then in the way that this kind of an inheritance diagram is drawn, all of the other specific Wnt's inherit those sites? Is that correct?
So yeah, it is close. So in this case, this generic Wnt has only one site, but that site, that feature has two members, and they are features of Wnt1 and Wnt2 specifically. So if you see this, and through through the link from Wnt to Wnt1 and Wnt2, you know that this maps to either Wnt1 that is phosphorylated at 21 or Wnt2 that is phosphorylated at 45.
Okay, makes sense.
Okay, and in Paxos we have facilities for kind of being able to navigate these guys relatively easily, but this is probably the most complicated part of BioPAX. But generics are never easy. So you can go to any pathway database, and generics are always difficult, both from a representational and semantic point of view. Finally, I kind of mentioned the template reaction and template reaction regulation, but I didn't mention what they are good for. So they are used to capture things like gene expression or translation, which are not necessarily stoichiometric biochemical reactions. They're essentially polymerization reactions with a template. That's the name. So what happens is that you can define a DNA region, which is a template for this reaction, and then you have the product, which can be an mRNA or a protein, and this whole thing can be controlled by a set of, for example, transcription factors or modulating proteins. Okay. Any questions so far? Okay, so the last structure that I want to talk about is pathways, which are lists of interactions, and in pathways are essentially demarcations of the large cellular map, and those demarcations are typically just done by the at the curator's discretion. So, for example, KEGG pathways are a lot larger than biopsych pathways, and there was even a paper about it, comparing their sizes and their predictive power, etc., etc. But my feeling is that when we are discussing about the whole big mechanism goals, we have to kind of pay particular attention to the fact that there's nothing particularly scientific about the boundaries of the pathways. I mean, they are typically artifacts from the way we think about the biological process rather than the biological processes themselves. But for what it's worth, BioPAX allows you to kind of create those demarcated regions as kind of taggings over the cell map, and what you can do is you can define the list of interactions. You can also define the order of these things happening. This order doesn't imply a temporal order because a lot of these pathways are cyclic. and there are multiple feedback loops, and etc. So temporal order is a tricky thing. In this case, we are really vague about the semantics of what the pathway order means. One particular use of pathways is that they can control and they can be controlled, different processes and pathways. So you can use them for representing things like the boundaries of your model. For example, if your model is leading to apoptosis, you might create a black box pathway which is empty. Name it apoptosis, tag it with a gene ontology term apoptosis, and make it control. So the last reaction of your model might control the apoptosis black box pathway, and this is one way of representing the events that is outside of the scope of your model. And some pathways databases, specifically NCI PID, exclusively does that. And pathways can also be nested. For example, reactome pathways can be like 4 or 5 deep sometimes. Questions? Okay, so these are the list of controlled vocabularies that we use in BioPAX. We try to validate the exports as much as possible so that they comply with those controlled vocabularies. The cellular locations come from the GO, subcellular compartment tree. There's something called relationship type vocabulary for representing different relationships. There's no accepted controlled vocabulary for this. Sequence modification vocabulary is used for representing like phosphorylation, acetylation, ubiquitination. For proteins, we are using the PSI-MI's PSI mode. For others, we don't have a set controlled vocabulary. The same thing with the experimental form vocabulary for representing things like like tagged proteins or other experimental forms. For tissue vocabulary, we are using BTO for annotating tissues. For cell vocabulary, we are using CL. And for evidence code vocabulary, we are using the PSI-MI for proteomics. For others, we are looking at several other options, but we don't have a set or accepted vocabulary. For phenotypes, we are using PATO, and for interactions, we are using the PSI-MI's interaction vocabulary. Finally, one thing that a lot of people discussed during the kickoff meeting is that BioPAX allows you to kind of extensively annotate evidence, provenance, organism, cell type, tissue, and external links. One important thing is that evidence object in BioPAX allows you to define an evidence code, which is captured in a controlled vocabulary, as I mentioned, an experimental form for representing things like fluorescent to tagged protein or etc., and also a confidence score that can be used to capture different levels of confidence in that reaction. Any questions so far? No?
I'm quite interested in the whole evidence code thing, and in terms of how the method, what kind of encoding of the methodology is you're using to talk about that, especially if you have a type of, if you have a typology of the types of experiments that you're talking about. So well, I'll follow up about that, and we don't have to talk about it here.
Yeah. So it actually is good. So we are using this vocabulary. So if you go to the PSI-MI, they have their own evidence code vocabulary, but that won't capture things like for example, that won't capture things different gene sequencing methods, for example. But it will capture all sorts of protein modifications and the protein pull downs, co-immunoprecipitation, all of these things.
Great, thank you.
But not necessarily, not all biological experiments that you can envision. If you would like to use a different kind of evidence code vocabulary or like a complementary set, I would love to discuss about how they would fit. And there's also one thing that I would like to mention is that there's no reason why we can't extend BioPAX for the purpose of big mechanism. And in fact, this is one of the things that I would like to kind of figure out if anybody would kind of come up with recommendations for BioPAX so that we can capture the big mechanism use cases that you have in mind well. Okay. So finally, just one kind of overview of how BioPAX kind of fits in with the other systems biology standards and related standards is BioPAX and PSIMI are very closely related to each other. And basically we capture most of the PSIMI structures for representing molecular interactions and genetic interactions. What BioPAX does on top of it is it can represent all of these biochemical events. And we have, for example, Paxos exports the BioPAX pathways into SPGN diagrams. So from BioPAX SPGN PD, there's a pretty robust mapping. The only thing that can't be visualized in SPGN is current to BioPAX generics. We don't have a good way of representing them, and I also think that you can use BioPAX to annotate SPML models and CellML models. Although a competitor in that domain is SPO, so you can kind of use, for this use case, you can either use SPO or BioPAX, and basically we use all these controlled vocabularies for annotating the BioPAX. Including, the sequence ontology, gene ontology, PATO, and both Miyazi and Mimics actually instructs what needs to be reported when you're publishing BioPAX models or SPML models. Okay, so one thing that I want to mention is that this is the big problem for us. For the use case where we are getting fragments from the natural language processing tools, also for the pathway databases, is that as these things are very complex, they're often and with a lot of unknowns, these are represented typically in slightly different forms and slightly different structures. So in this example, this is the same example that I gave in the kickoff meeting. Is these two reactions are talking about the same thing, although they have been annotated differently. They are both from the NCI-PI pathway database, probably curated by different curators, and as you can see, the base structure is the same, but there are also substantial differences. So we need to find a way to kind of align these. And regardless of the language that we choose or that you use, we will again face the problem, because we want to go from a redundant set of relatively simple statements in the literature to an integrated big mechanism. And this is going to be one of the key algorithmic challenges or programmatic challenges for us. Okay, so I want to kind of put an outline of what is needed for Big Mac from a BioPAX perspective, and then hopefully I would like to kind of initiate at least one round of discussion, and I'll try to keep notes about those as we discuss of like how we can use BioPAX and what else can be needed, or why for which use cases we can't use BioPAX, and if we can fix it, or if we can't fix it, what are the alternatives. And for me, these are the from the BioPAX perspective, these are the biggest questions. The one is the gap from TA1 to TA2. How do you convert NLP fragments to BioPAX, and what cannot be captured? For example, Andrei Zeski's group is suggesting a elaborate way to capture confidence. I think that Gali would like to chime in on that. But can we capture that type of confidence that kind of extends to multiple fragments of literature? Can we be able to capture that? How do we align and integrate, and how do we resolve conflicts? That's another big problem. And again, the last thing is the gap between TA2 and TA3. How do we convert to kappa, pathway logic, and SPML? And that's all I wanted to say. And I would like to kind of have your questions right now, and otherwise I would like to kind of start discussion on what you would need from BioPAX, and where do you think the biggest problems are? Where do you think that we can improve, or we can define APIs or tools?
Hello.
Hello.
It's Will.
Tim has gone.
Yes.
Okay.
Tim's up. Hey, Gary.
So, any questions?
Happening area, so.
So I think
Stop by.
one of the things that I thought was quite interesting is Paul has been mentioning the whole idea of the push model of publication.
Yes.
He's been talking about that, and I've been kind of trying to advocate and think about and talk about a nano publication oriented perspective on that. I don't know if people know about nano publications or what they've heard of it or what they think of that, but I think it's a good general principle kind of idea. And so I think that my focus is always going to be on experimental observations and what the evidence is. I mean, that's what I'm trying to provide for the community. But I envisage that BioPAX as a standardized approach to trying to express pathway data could be a nice way of a nice common lingua franca for us to be able to communicate with one another, but also as a model for publishing, and not just a lingua franca for use within the community, but a standard way of publishing information that could be consumed by people outside as well. So I think that would be kind of a really cool thing to put on the table as a use case for you guys.
We actually have been thinking about this for a while, and this again might be related to the. I think this also might be related to the big mechanism, and one thing that we would like to discuss. Let me quickly try to find a link. But we have a project called Factoid, and so let me find the link to the latest. Okay, so actually this is the Factoid. This is done with in collaboration with the Gary Bader's lab. Oops. So the link didn't show up right, but if you look at the comments section on the right, so there's the Factoid link, and this is exactly what we want to do. We want to be able to capture the BioPAX part directly from the author of a paper, and then publish it along with the paper. So you are probably suggesting even a more advanced use case where people don't publish papers at all and just publish the BioPAX fragment. And I think this is very interesting. But what we can also do is we can, as a part of the NLP pipeline that we are going to develop as a part of the big mechanism, we can try to kind of partially automate this process as well. So these are very, very interesting directions that I think we should all explore a little bit. And I think big mechanism is a good place to do that.
I would say add one thing to that. I think that you can't just publish the BioPAX fragments. You would need to include we'd need to figure out a way of publishing everything associated with the study, including the PDF, including a PDF file, including something that's human readable, including any kind of. So I think the BioPAX part is kind of like the summary diagram, right? It's like the piece of the fragment of knowledge that goes out into the world and
Yes. No, I 100% agree. I mean, BioPAX is only one facet of what can be captured electronically about a publication.
Exactly.
But that is the facet that I have the most experience and where my brain thinks. But you're 100% sure that you're 100% correct that there are many other aspects of a publication that can be captured.
Great. I think that's awesome. That would be really cool.
Okay. Brent, by the way, I already put the link to the Google Docs presentation. I don't know if you were able to get that, but
yes, I just got it.
Okay. So, any specific questions?
Yeah. So, can you hear me? This is Carolyn.
Yeah. Hi, Carolyn.
Hi. One thing which BioPAX, as I recall, doesn't support, but which should be easy, except that the databases don't support this. If you want to export to Kappa or Pathway Logic or whatever, in addition to the reactions, you actually need to postulate an initial state, because analysis typically starts with an initial state. I've made various algorithms to extract from the rules an initial state that would at least use all the rules, but it could be interesting to just provide a section that you could specify an initial state for your experimental analysis.
So when you mean an initial state, do you mean the actual concentrations of molecules and things like that, or do you mean?
It depends on which tool you're exporting to. So for pathway logic, it would just be what proteins are, what physical species or physical entities are present.
Okay.
In the initial state, it would certainly be useful to allow specification of concentration in different ways, whether it's really detailed molecules per milliliter, or just high, low, and medium. All of these different things I think would be really useful.
My first reaction to that is, yes, I agree. It is extremely important, especially if you want to do any sort of simulation or reasoning. But it also is highly context specific, right? Actually it is one way to specify the context, right?
Yeah.
So one thing that we kind of try to do in BioPAX is that we kind of first decontextualize everything and then merge them, and then try to reconstruct the context somehow. So that might not necessarily be the only way of doing things. And one effort in this direction, I think, is SedML standardization effort. SPML also can definitely capture the initial context, but all of these are really kind of is related to capturing the context problem as well, which we haven't addressed in BioPAX at all. So this is one thing that I also would like to kind of keep exploring and talk about, and I think we really need to talk about.
Yeah. So I mean, one thing to do is to have auxiliary documents, but documents which are compatible with the BioPAX, so that you would use the same vocabulary and standards, and you could have one BioPAX which gave you the reactions, but then you could have different contexts, but they would be easily linked.
I think that is an excellent idea. I am not sure how we can execute it, but that is something that we really should kind of keep thinking.
It's something that may emerge as we struggle with mapping all of these different representations between each other.
Yeah.
That's related to sort of some of the things I've been wondering about in the sort of the TA2 to TA3, going to executable models step, so because you're going to need initial concentrations, you're going to need rates, you're going to need to sometimes refine these generics into specific molecules, things like that, and I looked a little bit into this tool SBPAX for bridging BioPAX to SBML. That was one of the questions I had for EMEC in terms of wondering what's going on there. One of the things, as far as what I understand, is it's a bridging ontology that lets you go from BioPAX, which is for storing these facts, to SBML, which is more for running simulations, and in particular, it lets you make these mappings between different states that they're represented in BioPAX and into chemical species for use in these reactions that you specify in SBML. And one of the things that's nice about it is that it lets you save those mappings, so that if you tweak, if you want to get another chunk of BioPAX out and then render it as SBML, you can run it through that same set of mappings that you've created. I guess my question is, where does, because I mean I see that those types of tasks as things that we are going to have to do, whether we're mapping to pathway logic or to PySB or Kappa or what have you. Where do those kinds of tools stand? Are there things that we should try to use or try to write our own, or just kind of read what's been done so we can kind of conceptually leapfrog over some of the work that's been done, or try to reuse it from a technical standpoint, that kind of thing?
I think there are several people that that we would like to kind of pick their brain on this. And one thing that you mentioned is the SVPAX and a virtual cell group in general, because that's
Yeah.
something we've been doing for a while. And also you also mentioned in your email the Nicola Nolliver's groups and the the Andre Drager and others. They did several things. So VRK group spent a lot of time on similar problems, particularly related to rate constants and etc. There are definitely 4 or 5 major groups out there that try to tackle that problem, and I am also a newbie in that sense. I haven't done this or anything like this. I mean, particularly try to kind of capture the prior information about the the rates or about the initial conditions or concentrations. So we definitely kind of have to ask those people and see where their thinking are right now, because I also know that their position is also constantly evolving.
Mm-hmm.
But we might want to kind of email after this and try to kind of understand what they think for us to about what would be the best way for us to proceed. That is going from TA2 to TA3.
I guess along
Yes.
similar lines, the MedScan output. Anton Yuryov is not on this call, but he is very interested in looking at BioPAX. How do you proceed to go forward if we have different ways of modeling components of pathways that are similar but still distinct? What do you or everybody on the phone think is the best way moving forward to have these alternative representations and get to a point where we can at least import or export to each other's formats or something like that, or overlay them?
My feeling is that we basically have to do the standard procedure where with any other pathway database, where we first kind of start with the mapping, the either the database scheme or whatever scheme document that you have, and then try to kind of do some sort of initial course mapping, and then we can kind of try our hands in one pilot pathway conversion and then look at the problems, and then from there it typically is an evolving process. So we might actually have to extend BioPAX if some of the features that you're representing can't be represented by BioPAX. But that is definitely one exercise that I will be very interested in doing and have done in the past. So it all starts with the mapping schema. Another useful thing might be kind of have a common document repository for all of these mappings or schemas for the big mechanism. So that is another place, because otherwise we'll have to do n-square type of dialogues and conversions and do the same exercise again and again.
Yes, that's
but
okay, good.
but my short answer is that if you guys would like to email me with some initial document, I'm really happy to start working on it, and then we'll try to give you a response ASAP.
Yeah, and you're mapping, I would assume, the RAS pathway or some form of the RAS pathway, or I mean, you could do it the other way around.
We might do RAS as well. I mean, but yeah, sure. We might do RAS as well, but any other pathway will do.
So one thing we could contemplate, if you go and ask some of these people who do the actual kinetic modeling, and you say, where do you get your rates and concentrations? And some of them have pointers to papers. Many of them are rational, informed guesses. It might be useful to try and collect somehow a knowledge base that gives you plausible concentrations for different things in some reasonable collection of cell lines.
We can definitely help with a lot of that kind of stuff. In some of the typical cell lines that we've worked with, we've measured some of these proteins. Well, in the other pathways that we model, but that's something that we have experience with, and also in terms of kind of coming up with reasonable guesses about rates from prior knowledge based on the type of molecule and things like that.
Right.
So
That would
there's
be
interest
great
in that.
because that could help automate the passage from BioPAX plus initial state to a kappa model or something like that.
Yes, exactly. I mean, those are the one of the things that we're hoping to do is, so when you go from a set of facts or the literature or pathway databases to a mechanistic model, there's a series of assumptions that you make, or you kind of draw the line when there's some ambiguity in the literature. You come down on one side or the other, and then you make inferences about whether certain types of binding are competitive or not. And those are the types of things that generally go on. That's part of the art of modeling, and I guess one of the things we're hoping to do is make that process more explicit and manageable as a kind of procedural assembly process. So yeah, that's definitely something that we're hoping to work on.
Cool.
I don't know if I can't really see if the showcase plugin is working, but I also put a link to the sub-ERK both into the comments and in using the showcase. I don't know if you can see it, but that is one group that actually spent a lot of thinking in terms of capturing those experimental conditions and parameters. I'm not sure about the the combined context of prior context or starting context. I don't know what the right term for it is. I don't think they're doing this, but in terms of experimental conditions and rate equations and things like that, this is the group that I know that spend the most time and thinking on this. So it might be worth exploring.
So I'm not
I think
seeing
I also
any...
posted this, but as you mentioned, Sophia Ananiadou, Sophia Ananiadou and Larissa Soldatova also, of course, have both developed models of certainty and evidence levels and such. And so it might be good to have a separate call with everybody who's interested in modeling evidence.
I think that's a very good idea.
Just on that.
Maybe we should have like a separate hangout for how to capture evidence and confidence.
Yeah.
Caroline, were you saying something?
Gully,
oh,
do you think maybe you...
Sure,
I
that
can
sounds like a good idea.
Maybe Gully and I can talk, and one of us can perhaps kick this off.
Yeah, sounds like a good idea. Caroline, were you saying something?
Oh, I was just saying that whatever this sidebar that you have is not showing up on I'm using my iPad. Is there a place that you've stored all this stuff that we can get it later?
Yeah, sure.
Yeah, or maybe send around a link or something.
Yeah, I'll do that.
Yeah, great.
Anything else?
I think the devil is in the details. Once we start implementing mappings.
There's so much we can do within a 1 hour call, right? I mean, when we start when we start implementing things, we'll start hitting all these problems. But hopefully this was useful for kind of lay out the what is out there in terms of BioPAX and what can BioPAX represent, and then B, what the other directions that we should start following and kind of thinking about.
Absolutely.
Okay, thanks for the participation. So sorry for those who couldn't make it, but hopefully the stream is useful. So catch you in the next meeting or in the next hangout.
All right. Thank
you. Have a good
you.
one.